Hello, welcome to the last live of the year inside the Ultimate Review Packet. So this live is completely exclusive just for students who purchase the Ultimate Review Packet. So hello, and thank you so much for your purchase. I greatly appreciate it, and I wanted to make sure that I gave you some exclusive content in order to be prepared for tomorrow. And I cannot believe that it's tomorrow. So obviously, you're here because of the URP. Also, the podcast may have been how you found the URP, and we have our YouTube resources, all of which are going to be helpful for you as you prepare for the exam. So there's actually going to be a study guide that will help you go through this presentation. It's in the Ultimate Review Packet. You can see the link right next to the link for the live. That is going to help you just follow along, grab some of the information from these slides. We're going to use the chat, right? So if you have questions, if you want to have a topic request that you'd like me to go over, you can pop it, and I'm going to hit as many as I possibly can. Just please be respectful and help each other out as you go. So as we always say, let's zoom in. AP Bio, it's super difficult. Don't be defeated, just be challenged. So the exam tomorrow is going to be 3 hours long. We're going to split in two pieces. 90 minutes for the multiple choice. We have 90 minutes for those FRQs. They're going to test a variety of skills in both sections. Now, you already know that the task verbs are bolded, and that's going to equal the points. So you want to make sure that you answer the points, answer the task verbs in order to earn those points. And that really leads us to look at how the point values are accounted for over time. So we're going to do a quick pop quiz, true or false. A point earned in the FRQ question is worth more than a point earned for a multiple choice question. So take a moment to think, how many questions do I have in each section? And that's going to be a clue to the response that we have. So it's absolutely true. 60 questions in multiple choice means 60 points. The points in the FRQ section, because there are fewer of them to earn, it's about 1.7 value. So especially true that you don't want to leave things in the FRQ section blank. So we're going to do some practice with how to break down an FRQ, specifically for the redesign that you will see tomorrow. So if you've been using old FRQs that are published by the College Board, there weren't very many that were restructured. And so there wasn't a lot of valid practice to the format that you're going to see tomorrow. So if we look here at the breakdown, we can see that my introductory paragraph about the polycystic kidney disease, this is from the 2021 free response questions that they reformatted. This represents question number 1, which is a long FRQ. You can see that it's subpart A, and then I have I and little I, so 1 and 2. These breakdowns are going to allow you to only know that there's a certain amount of text you have to respond to, and the rest of it is not going to be relevant to answer this question. Now, I've been saying it for a while, but part A is really prompt independent most often. And so it might be related, but it's typically content adjacent. What we see in our scoring guides, I do want to point out, there's variety. You also have things at times written in parentheses, and that's going to mean that you don't actually have to say those particular phrases. Whenever you see a slash, that means that it'll accept either or for the response. So this is the second part of that FRQ. We're not going to go through the whole thing, but I just wanted to show you one more time how the breakdown would be shown. This particular is going to be reflecting back on the graphs when you are identifying a dependent variable, justifying the use of a particular concentration. Those are all going to reflect back on information directly above it. Now, it is possible that you will need to go all the way up to the first part of the prompt and not just into this immediate section. to answer these components. But what it does imply is that there's nothing beyond this point, no text beyond this point that you will need to reference in order to be successful on the exam. So when it comes down to kind of like the big tips for FRQs, what you want to be aware of is really address the bolded task verbs. You don't want to just restate the prompt. Restating the prompt or defining bio terms that the readers know is really just a waste of your time, and there's not going to be any points for definitions. Quality over quantity. I've had students that write a single sentence that are completely valid in their response and earn all the points. I've had students write 15 sentences, and by the time they get to sentence 6, they've just taken away points, right? They've negated something that they had said previously, and that's not going to be very helpful either. Don't use any pronouns. You saying it changes, or it is different. Well, first I'm going to say what is different, and then I'm going to say different how. So both of those pieces are going to imply that you have to be more specific. So pull a term out of the prompt, pull something out of the graph specifically. You also always want to use full sentences. Now, it's not actually a College Board requirement that you do this, but as a teacher, there is a lot of validity to help your thought processing to using full sentences. And it likely will just help you catch yourself if you're going to make a mini mistake. And so that that ability to just say the independent variable is will allow you to double check that you are in fact selecting the independent variable. The key thing about FRQs though is you can answer them in any order, including the subparts. So with the example I just showed you, how it had A, 1, 2, 3, you can answer it A, 3, 2, 1. If that is the way in which you knew the answers and the fastest way for you to earn points. We'll find it as long as you have it labeled. You do have to use pen on the FRQs because all of your booklets are scanned, and the pen will show up a lot better on the scan because they're graded digitally. This is true for the graph as well, right? If you are nervous and you want to use pencil on your graph, you should go over it in pen, assuming that you have time, because it's going to make sure that you can, it's most legible for the readers is to use pen. Now, if you are, as it's the night before the exam, going like, okay, what should I do to spend my time? Like, where is the time well spent? Unit 3, Unit 6, and Unit 7, because that's going to be the greatest percentages. And so if you're a little foggy on things from Unit 1, don't let that bother you. Just kind of let that go to the wayside because it's only 8 to 11%. And at this point, you got to play your odds. Spending more time on the areas that are most represented or should be most represented on the exam tomorrow is going to be your best bet. But if you have absolutely no clue, choose the answer that has to do with energy efficiency, gene expression, or increased variation leads to evolution. Those are the themes that we see through all of the big ideas that go across the 8 units. And this is what that looks like. So those types of responses show up the most because they're the big ideas that are going to be most prevalent through all of the 8 units. And just be careful not to fall into the trap of picking the answer that kind of sounds most sciencey, because there's going to be terminology that you know in the wrong answer choices, not to confuse you, but to just test whether you can dissociate the difference between the correct response and just science jargon. So we're going to do some practice with math and questions to kind of go through some of the big themes and concepts. I haven't done a lot of math in the URP a ton, and I feel like it would be a useful thing for us to review right now. So we're going to start with kind of the overview. Remember that the AP Bio formula and equation sheet is going to be on you for the exam. You can't rip it out though, and that can be a little frustrating. So if you have more than one writing utensil, if you brought an extra pencil, use the pencil as a bookmark for the page that has the formula sheet. It's going to allow you to flip back and forth a lot faster. So if we look here at a genetics practice, this is completely imaginary, made up organism. Let's assume that the traits for tall and grumpy assort independently, meaning that they're not linked genes. Show the expected genotypic and phenotypic ratios of a cross between a tall, pleasant zoodle and a short, grumpy zoodle. Homozygous recessive completely for the short, grumpy zoodle. And so I personally prefer to foil my gametes to determine the Punnett square size that I need. This is not good, although it's a dihybrid problem. A 16-box Punnett square is just completely unlikely. Or you could take each of the crosses and take them independently. But here we have genotypic ratio being the actual expression of my alleles. Phenotypic ratio is going to be the physical characteristics once those alleles are translated into protein. And so the response here would be 50% and 50%. When I foil my parental genotypes, I'm going to be deciding and figuring out what gametes they could produce. So let's talk about water potential. Water potential, literally the potential for water to move based on pressure and solute potentials that we see. The solute potential of a solution is negative I CRT, and all of this is copied directly out of the formula sheet. So this is going to be something that you can rely on. You don't have to memorize what those letters stand for. So if we're calculating the solute potential of a 0.2 molar salt solution at 25 degrees C, we need to be mindful, what is the ionization constant of salt? Well, salt ionizes into sodium and chlorine, and so that ionization constant is going to be 2. 25 degrees Celsius, I look over at temperature, but I see my temperature has to be in Kelvin. So I do need to convert my temperature and make sure I add my 273 to get my final answer here, which is in bars. If the concentration of salt inside the plant cell is 0.25 molar, which way will the water diffuse if the cell is placed in the 0.1 molar salt solution? So here we have a difference of concentration. We're referring to hyper and hypotonic. And when we use those terms, remember, it's going to be a description of the solute concentration. Water, on the other hand, follows its own concentration gradient. I always recommend doing some type of diagram or sketch when I'm trying to determine the movement of my solutes or the movement of my water. In this case, I'm going to have water flowing into the cell, following its own water potential gradient. Now, this is a plant cell, so we should have turgor pressure increasing as my vacuole fills with water. So, taking a quick halftime break. What's next? What's going on? More AP classes, graduation, maybe college. Are you going to have a particular major? Either way, I hope that the AP class or classes that you've taken have served you well, including AP Bio. But they do say it's good for your college resume, right? I would argue it's good for much, much more than that, that helps you out beyond college. All right, our next equation that we're going to be looking at is Hardy-Weinberg equilibrium. So remember that Hardy-Weinberg equilibrium is really a theoretical model for non-evolving populations. Non-evolving meaning it doesn't meet any of those 5 requirements for evolving populations that we've discussed previously. So here, if we have a mouse population of 100, 20 represent the homozygous recessive white mouse. We want to know what is the frequency of the dominant allele and how many mice are heterozygous. Now, we don't know any information about what that dominant allele represents, but we don't need to answer this question. P is going to be my dominant allele. Q is my recessive. All of my equations here will add up to 1. And so always, always, always start with the recessive because it does represent a phenotype exclusively that you can extract the value of Q from. And so here, Q is going to be 0.45. And by subtracting from 1, I can then determine my value of P, the dominant allele. But the question then asks how many mice are heterozygous? Heterozygous being represented by 2PQ And so using those values to plug in, I can see that 50 mice here are going to be heterozygous. All right, next up for the math, chi-square hypothesis testing. Once again, pulled off of the formula sheet, all of which is represented and defined for you so that you can not have to memorize what these stand for, right? Degrees of freedom is a number of choices, n minus 1. My p-value, I've really seen only 0.05 used on the AP Bio exam, and that represents a 95% confidence. And so we're going to do the same in our practice problem. So if there are 100 heterozygous males that mate with 100 heterozygous females, the observed outcome from this experiment is 28 homozygous dominant, 56 heterozygous, and 16 homozygous recessive. Calculate chi-square for this experiment, okay? Now, we haven't yet really challenged us to do anything with that value. Typically, what chi-square will say, do the expected results, observed results match what is expected, or is there some type of external factor influencing our results that we should be mindful of? So the best way to go about these problems is really to make a chi-square table to organize our data, O minus E, O minus E squared, and then again all over E. You're going to sum these values to come up with your chi-square. The reason why we have 2 degrees of freedom is because my choices, my 3 different genotypes, are going to represent N. N minus 1 gives you degrees of freedom, and that's why our degrees of freedom here is 2. Looking at my chi-square chart, my p-value of 0.05, I'm going to compare my calculated value of 4.32 to 5.99. I pulled it directly off the chart. Now, this is less than my critical value, and so by the point of expression, I would say that the null hypothesis is not rejected. And that's, I know it's a little odd to say that or supported. There isn't anything going on here that says there's external influences on my results. Moving on to Simpson's diversity index. Again, we see a summation, a sigma, looking at the total number of organisms of a species relative to that of a whole. We are attempting to define species richness within a particular area by using this equation. So instead of using organisms, we're just going to use a deck of cards because the numbers work out the same way. So if you had 10 cards randomly sampled from a deck, 3 were spades, 2 were hearts, and 5 were diamonds, what would be the diversity index of that population? And so the way that we're going to work through this problem is going to be to take them individually, since I have my summation. Taking n lowercase n over capital N and squaring it. So 3 over 10 squared, 2 over 10 squared, and 5 over 10 squared. Then I'm going to add my values to get 0.38, subtract that value from 1. And so we typically would be comparing this diversity index to either a known value from a previous sample to see how the diversity may have changed, or to another population. And then there might be a question about, which population would be more resistant to an ecological change. So before we move on from math, I do want to point out a couple things. Units do matter. So do be aware of that. They're typically pulled right from the prompt itself. So use units when you can. I always get a question about rounding. Rounding isn't advised, but you don't want to write numbers in perpetuity. And when you have to take values off of a graph, you're estimating. So typically in the FRQ scoring guide, there will be a range of numbers that is accepted that your answer has to fall within. So common misconceptions of biology. I really wanted to go over just some common things that might be said, or things that might be cobwebby from freshman year bio or a bio that you may have taken before AP, so that we can kind of talk out what is different in this as compared to the others. All of these statements are not valid. And the question here is, okay, like what's wrong with them to make them invalid? I'm going to make the screen a little bit bigger so we can see them. So I'm going to go away for a minute. All right, so these are all incorrect. The question is, why are they incorrect? The first states enzymes are necessary because they cause reactions to happen. They don't. They may make it more likely for a reaction to happen, but reaction rates can be influenced by a lot of factors. Enzymes are just one of those factors. When we look at reactions, they are typically grouped into spontaneous and non-spontaneous, endothermic and exothermic, and it really depends upon the value, like how much activation energy is required for the reaction to occur. But all reactions do require some sort of input of energy. All right, the second statement says cellular respiration is only done by heterotrophs because autotrophs can make their own energy. First of all, nobody's making energy because energy cannot be created or destroyed. It's the law of thermodynamics. If I am an autotroph, hooray for me, I can produce my own food by capturing energy either from the sun or from chemicals, right? So we have photosynthesis and we have chemosynthesis. But once I have this glucose or this food, I still have to do something with it, right? The cell doesn't speak glucose. I have to convert into an alternate form, ATP being that form. So autotrophs are going to have the ability to do both photosynthesis and cellular respiration or chemosynthesis and glycolysis and fermentation, something like that. They have to do both. Plants only use water as a means of keeping their cells full and holding the plant itself upright. Not correct. In fact, water is so important to plants because without water, photosynthesis wouldn't happen. So, water is going to be split by the process of photolysis and donating electrons to the electron transport chain. So, as electrons move down the chain, they are replaced with electrons from that split water. Dominant alleles are found at greater frequency than recessive alleles in populations. I can see how this would seem correct, and there are circumstances in which this is true, but it has nothing to do with the fact that they are dominant. Dominant alleles are termed such because they mask the recessive allele, not because they're better or they have greater survival rate or have an evolutionary advantage. It has to do with the way that the proteins are expressed. And so you will find this to be completely varied in terms of which alleles are going to be selected for within a population. That's going to change over time as the environment changes. Genotype always equals phenotype. So also not true, right? So we have differential gene expression. We can have environmental factors that also influence how those genotypes are expressed. And so whether it be through environmental chemical or just the fact that you are exposed to sunlight, and so there are going to be different physical expressions within your cells, all of that is not going to be exclusively due to your genotype. When the environment changes, all species living in it will change to adapt to it. Well, that would be lovely, but adaptations are required over long periods of time, and they're based upon, yes, the environment, but also the organisms within it and the competition that exists. And so you're either going to adapt or die in the environment. Those are your two choices. And by choices, I mean those are the two outcomes. But no, not everything is going to adapt. If that were true, we would not have had extinctions. All hormones have the same types of effects on cells, no matter what they are made of. So, hormones are typically either lipid hormones or protein hormones, and that will influence where they bind. So, proteins have extracellular binding sites, lipids have intracellular because they are nonpolar. But that nonpolar is going to be influential in terms of the cascade effect that happens. So, reception, transduction, response, those are our three stages of cell communication. And cell communication and the effects of that communication really do vary depending upon the type of hormone that it is. And so, no, hormones are very specific. Lastly, all populations will increase continuously regardless of outside factors. So, this is referring to exponential growth model. And there are many populations that do experience exponential growth until some sort of limiting factor comes into play. It might be food, it might be shelter, it might be mates. But the population is going to reach carrying capacity, and we're going to enter a logistic growth model. If a factor were to change again, the population could go back into exponential growth, but it tends not to be in perpetuity. So these are all incorrect misconceptions. All right. So I am a member of a large community of AP Bio teachers, and I've asked them to share some words of advice with you on the eve before the AP exam. Like, what would you tell your students, or what do you tell your students in order to kind of like get the jitters out and find the most success? Some of it seems pretty obvious, but I thought it would be useful to share with you too. Don't leave anything blank. So if it is the multiple choice and you are in the last, let's say 5 minutes, and you still have 10 questions left, bubble, 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 bubble. Don't leave anything blank in the multiple choice. I know you're just guessing. You didn't even get a chance to read the question, but you might just get lucky and get some of those correct. In the FRQ section, it's a little harder not to leave anything blank. So if you're at the last 10 minutes and you have an entire question you've not even gotten to, scan the question and see if there's any like freebies, meaning is one of them just asking for a variable off a graph, because that takes no time to pull off. Or if you don't know which variable is which, just pull one of the axis labels off. You might get right. If it asks you to make a prediction, that's kind of a freebie answer, right? The options are increase, decrease, and stay the same. So pick one. Lots of ways not to leave things blank in both the multiple choice and the FRQ section. I like this one because it's a lot like evolution. APs are like running from a bear. You don't have to do well, or you don't have to be faster than the bear. You just have to do better than other people, everyone else, right? So the scores are kind of put relative. It's not going to be a curve, but it is a cut score. So the score, the number of points you need to earn changes from year to year based on the difficulty of the test. So let's just say that last year was 80% correct to get a 5. The year before was 75. I don't know the actual percentages because it's not public information, but it does vary from year to year. So don't worry about having perfection. You don't have to earn 100. You just have to do your best. This one is a little bit more specific, but the answer has to be figured out often. It's not just going to be overt or something you just happen to know, except for maybe for part A of the FRQs. But the rest of it, you have to pull data, pull terms, pull phrasing from the prompt itself. So just know that it's not typically going to be knowledge you take from a place that's in the depths of your brain. It's going to be something you synthesize based on information from the prompt. Speaking of ATP, answer the prompt. If you kind of have like bio vomit all over the test, and it's just going to be like everything you know about this topic, the greater chances are that you're going to negate something that you said that was correct later on down the prompt, and that just would be a waste of your time, and you're going to lose points. And this one I think is the most important one to remember the night before the test, because you are so much more than a test score, and you know so much more biology than one test can show, right? The proportion of things they can possibly ask you tomorrow is only a fraction of what you've learned this year. And so with that, get sleep, eat breakfast tomorrow, hydrate, Definitely breathe. It's just a test, and you've prepared so hard this year going through the ultimate review packet, going through all of the things on YouTube, going through the resources your teacher has given you. So just trust that, you've done your due diligence and now you're just going to try your best because that's kind of all you can ask for. So to recap, we have 8 units, 4 big ideas, 6 skills primarily. The exam has 2 parts. It's 3 hours, which is a long test, but you can do it. And so I absolutely believe in you. And I want to say thank you so much for supporting me this year with your support of the packet, for watching videos over on YouTube. If you found any of this helpful, it would mean the world to me if you could tell someone taking AP Bio next year to come check out my resources. This is our first year of the AP Bio Ultimate Review Packet. It's our first year over on YouTube. So these are all new resources that have been produced within the past year. And so I hope you found them helpful. I hope they gave you confidence to be your absolute best tomorrow. And I wish you all the best. And I will perhaps see you next recap.